Vagus nerve as modulator of the brain-gut axis in psychiatric and inflammatory disorders, Part 2. This was published in Frontiers in Psychiatry, published March 13, 2018, the National Library of Medicine, National Institute of Health. Vagus nerve stimulation. Vagus nerve stimulation is a medical device that is routinely used in the treatment of epilepsy and other neurological conditions. VNS studies are not just clinically but also scientifically informative regarding the role of the vagus nerve in health and disease. Vagus nerve stimulation works by applying electrical impulses to the vagus nerve. The stimulation of the vagus nerve can be performed in two ways. A direct invasive stimulation, which is currently the most frequent application, and an indirect transcutaneous non-invasive stimulation. Invasive VNS, it requires the surgical implantation of a small pulse generator subcutaneously in the left thoracic region. Electrodes are attached to the left cervical vagus nerve and are connected to the pulse generator by a lead, which is tunneled under the skin. The generator delivers intermittent electrical impulses through the vagus nerve to the brain. It is postulated that these electrical impulses exert anti-epileptic, antidepressive, and anti-inflammatory effects by altering the excitability of nerve cells. In contrast, to IVNS, the transcutaneous VNS allows for a non-invasive stimulation of the vagus nerve without any surgical procedure. Here, the stimulator is usually attached to the auricular concha via ear clips and delivers electrical impulses at the subcutaneous course of the afferent auricular branch of the vagus nerve. A pilot study that examined the application of VNS in 60 patients with treatment-resistant depressive disorder showed a significant clinical improvement in 30 to 37% of patients and a high tolerability. 5 years later, the stimulation of the vagus nerve for the treatment of refractory depression was approved by the U.S. Food and Drug Administration, or the FDA. Since then, the safety and efficacy of VNS in depression has been demonstrated in numerous observational studies, as can be seen below. In contrast, there is no randomized placebo-controlled clinical trial that reliably demonstrates antidepressive effects of VNS. The neural mechanisms of VNS. The mechanisms by which vagus nerve stimulation may benefit patients non-responsive to conventional antidepressants is unclear, with further research needed to clarify this. Functional neuroimaging studies have confirmed that VNS alters the activity of many cortical and subcortical regions through direct and indirect autonomic connections via the NTS. The vagus nerve has structural connections with several mood-regulating limbic and cortical brain areas. Thus, in chronic VNS for depression, PET scans showed a decline in resting brain activity in the ventromedial prefrontal cortex, which projects to the amygdala, and other brain regions, modulating emotion. VNS results in chemical changes in monoamine metabolism in these regions, possibly resulting in antidepressant action. The relationship between monoamine and antidepressant action has been shown by various types of evidence. All drugs that increase monoamines, such as serotonin and dopamine, in the synaptic cleft have antidepressant properties. Accordingly, depletion of monoamines increases depressive symptoms in individuals who have an increased risk of depression. Chronic VNS influences the concentration of 5-HT, NE, and DA in the brain and in the cerebral spinal fluid. In rats, it's been shown that VNS treatments induce large time-dependent increases in basal neuronal firing in the brainstem nuclei for serotonin in the dorsal raphe nucleus. Thus, chronic VNS was associated with increased extracellular levels of serotonin in the dorsal raphe. Several lines of evidence suggest that NA is a neurotransmitter of major importance in the pathophysiology and treatment of depressive disorders. Thus, experimental depletion of NA in the brain led to a return of depressive symptoms after successful treatment with NA antidepressant drugs. The LC contains the largest population of noradrenergic neurons in the brain and receives projections from NTS, which in turn receives afferent input from the vagus nerve. Thus, VNS leads to the enhancement of the firing activity of NA neurons and consequently an increase in the firing activity of serotonin neurons. Thus, VNS was shown to increase the NE concentration in the prefrontal cortex. The pharmacologic destruction of noradrenergic neurons results in the loss of antidepressant VNS effects. In the case of DA, it's been shown that the short-term effects, 14 days of the long-term effects, 12 months of VNS in treatment of resistant major depression, may lead to brainstem dopaminergic activation. DA is a catecholamine that has a large extent is synthesized in the gut and plays a crucial role in the reward system in the brain. Further, beneficial effects of VNS might be exerted through monoamine-independent way. Thus, VNS treatments might result in dynamic changes of monoamine metabolites in the hippocampus, and several studies reported the influence of VNS on hippocampal neurogenesis. The process has been regarded as a key biological process indispensable for maintaining normal mood. Serotonin is also an important neurotransmitter in the gut that can stimulate peristalsis and induce nausea and vomiting by activating the vagus nerve. In addition, it's essential for the regulation of vital functions such as appetite and sleep and contributes to feelings of well-being. To 95%, it is produced by enterochromaffin cells, a type of neuroendocrine cell that reside alongside the epithelium lining the lumen of the digestive tract. Serotonin is released from enterochromaffin cells in response to mechanical or chemical stimulation of the gastrointestinal tract, which leads to activation of 5-HT3 receptors on the terminals of vagal afferents. 5-HT3 receptors are also present on the soma of vagal afferent neurons, including gastrointestinal vagal afferent neurons, where they can be activated by circulating 5-HT. The central terminals of vagal afferents also exhibit 5-HT3 receptors that function to increase glutamatergic synaptic transmission to second-order neurons of the nucleus tractus solitarius within the brainstem. As a result, interactions between the vagus nerve and serotonin systems in the gut and the brain appear to play an important role in the treatment of psychiatric conditions. Vagal-related treatment of depression. Basic pathophysiology of depression. We'll conclude this and I'll start this in part 3.